Welcome back. I'm going to continue this discussion of self-organization, but now get a little bit more into the details. And I mean, what I was trying to do in the last talk was really give you my sense of where molecules become living systems. But now we're going to try and really look a bit more at how do actual molecules do that. And I'm going to use this example of the meiotic spindle here. Again, here are some outline and concepts that I won't read out now. So the examples I'm going to use are all in the context of cell division, and I thought it would be nice to start this with the very first description of cell division by Fleming in 1882, more than 100 years ago. And he, looking at fixed and sane salamander cells, drew these absolutely exquisite pictures, and you can see here, these lines here are the mitotic spindle. And Fleming realized what the job of the spindle was, which was to split the chromosomes into two groups. You can see them here being split at anaphase so that the two daughter cells get identical numbers. What Fleming, of course, didn't know is why is it so important to split the chromosomes in two. Now we know that's where the DNA is. It's important that every cell in our body get an identical copy of the genome. So the mitotic spindle has to very precisely split the chromosomes in two. Here's a slightly more updated view of this. This is a movie of a tissue culture cell dividing. The chromosomes are these dark objects in the middle. And you can see here they are lined up in the middle. And then in this movie, you'll see them be split in two. And the forces are coming from the mitotic spindle. This is a movie I took, actually, 1988. I took myself when I got my first microscope in my first lab in San Francisco. I'm very fond of this movie. But I think it shows the process rather nicely. And then after the chromosomes, this pinching in is called cytokinesis. That's where the cell actually divides in two. In this image, I've superimposed on the phase contrast. This is not the same cell. This is a much later image, but it's the same cell type. You can see here the chromosomes stained red, and the microtubules that are exerting force on the chromosomes stained green. This is an image by Julie Kahneman and Ted Salmon, Microtubules and DNA. And just a little bit more terminology. I'm going to use the word spindle pole for this point where the minus ends, the microtubules are polar, all the minus ends gather together here at the spindle pole. And in some types of spindles, you have a centrosome here, a nucleating site. And then this point here, the plus ends, some of the microtubules, a subset actually, meet chromosomes at kinetochores. This is where a lot of the force is generated to actually move the chromosomes. So I'll be using those words. I'm not in this talk going to focus on how chromosomes are segregated, how that's made so accurate. That's a fascinating topic, and many people doing beautiful work on that. And I'm going to stay more on the question of how you self-organize this shape and how it gets its lifelike properties. The system that my lab has used a lot of this are eggs here. You can see a frog egg dividing, and these frog eggs come from the Xenopus laevis, the African clawed toad, which is an amphibian that doesn't seem to mind growing in the laboratory. They live for ages in the laboratory. They aren't hurt. They lay eggs, and then we have them recover, and then they lay eggs again a few months later. And you can see it divides beautifully. And so this is a system I like very much for studying cell division. And we study one particular type of spindle in these eggs, which is actually the meiosis II spindle. And you can see it in this image here. This is an unfertilized frog egg, this big sphere. And the meiosis spindle is this teeny little point up at the top there. And I really like this image. I have a huge one. Martin Woer, my ex-student who took this image, blew up one of these to be 4 feet in diameter, and it's hanging outside my office. It's sort of symbolic of my life. Here's this world of the cell, and I've spent far too many years perhaps studying this one tiny little point here up at the top. But if you zoom, and let me also emphasize the size here. So the frog egg these Xenopus eggs are 1.2 millimeters in diameter, which really emphasizes the challenge of spatial organization of such a large egg. Here's a zoom in to the meiotic spindle here. It's about 30 microns in diameter. You can see the DNA in the middle. The job of this little spindle here is to complete female meiosis. So when the egg is fertilized, it separates the two copies of the maternal genome, splits them into two. One copy is thrown away, the other copy meets the paternal genome, which comes in, a sperm would come in, something like this, and the sperm nucleus moves into the center and then meets the one half of the female genome. So, but the job of this spindle is to essentially throw away one copy of the maternal genome and keep the other to make it haploid. Let me just a little bit of review here. I've shown you a somatic spindle dividing that PTK2 spindle. Those spindles have centrosomes here, but the spindle we're going to focus on, these egg meiotic spindles, are organized a little differently. They don't have centrosomes. They're sometimes called anastral spindles, and other organisms make spindles that way. For example, higher plants lack centrosomes, and they have spindles that look a lot like these egg meiotic spindles. Now, Xenopus eggs, although they divide beautifully, they're large and opaque, which makes it hard to visualize what's going on with a microscope. And I'm sure it's evident to everyone by now that the favorite thing in my lab, my favorite thing to do in biology, is to peer at things through a microscope as they happen. And it turns out we can get away around this problem of lack of opacity of the egg by preparing an extract from the egg and using that extract to recapitulate biological processes either in a test tube or under a microscope slide. And when you make an extract, in addition to being better for microscopy, it's great for biochemistry. You can add and remove components and do many kinds of manipulations that aren't possible in a living system. And so this is an experimental system that's very dear to my heart. The female frog lays eggs. She's injected with hormones, lays eggs, and then when that's done, goes back to the colony to recover. We collect those eggs, we wash them, we pack them in a centrifuge tube to kind of squeeze out all the pond water that would otherwise dilute it, or the buffer we've washed them with. Then we crank up the centrifuge to 10,000 G. That puts a lot of force that crushes the eggs and also separates their internal content here. So the yolk is down at the bottom, there's some fat up at the top. And here is the kind of liquid part of the egg, the cytoplasm, and this is essentially undiluted. This is a kind of 1x cytoplasm. This is a method developed by Andrew Murray when he was in Mark Kirshner's lab, and my very first PhD student, Ken Sarwin, implemented this system for studying meiotic spindle assembly. This extract, as I say, it's really cytoplasm. It contains abundant organelles, mitochondria, ER, etc. It also contains glycogen, an energy reserve, ribosomes to make proteins. Really everything in the cell except nuclei. It's metabolically active. It actually consumes about 1 millimolar of ATP a minute, which it remakes. And the mitochondria deplete oxygen, which turns out to be very good for fluorescence imaging because oxygen tends to bleach fluorochromes. And finally, the cell cycle state can be precisely controlled, which is essential for these experiments. So I wanted to walk you through a very typical extract experiment, and this is a kind of experiment hopefully I'm going to be doing later this afternoon in Woods Hole. So the first thing is to prepare this extract, and on a typical day we would, for example, collect the eggs before lunch, and the extract would be ready after lunch, and you can store it on ice for up to 8 hours. You can freeze and thaw it, but it doesn't work so well, so we always prefer to do it fresh if we can. If you want, you can remove a protein of interest with an antibody. We almost always add fluorescent proteins so we can do imaging. A lot of the experiments I'll show you add tubulin labeled with rhodamine so we can see microtubules growing. We typically add a nucleus, or I'll show you some experiments with DNA on beads to trigger spindle assembly. You can add a drug, an inhibitory reagent, something like that. Then typically you take a few microliters, put it on a slide, drop a cover slip on top, seal it with wax, and then image by live fluorescence microscopy. And then a very essential part, when you get a good movie later, not the same day, you have the movie stored, and you would do quantitative image analysis to figure out what happened and quantify what you've perturbed compared to wild type. And this whole procedure, from here on down, takes less than an hour. One reason I love this system is you can come to the extract, at 1 o'clock in the afternoon with some big theory, and by 3 o'clock, you've already disproved that theory and you're all despondent. You try a few more things, and then by 7 o'clock in the evening, you've got some new theory and you're working crazily into the night to figure out if it's right. So it has this very fast pace, which I just absolutely love. Of course, you have to make a bunch of reagents beforehand to allow these experiments to go that fast. And we can recapitulate at least 2 kinds of microtubule assembly. I'm really only going to talk today about these meiosis II spindles. We're recapitulating that tiny little spindle that lived at the top of the egg. I'll show you one movie. We're also increasingly working on microtubule asters, which is a new direction for my group. They are functional, these spindles. I'll show you one movie of the spindle actually segregating chromosomes. The chromosomes aren't shown here, but in red, I hope we'll be able to see them. These little red dots here are kinetochores. And to trigger anaphase, we add some calcium. When the sperm comes into the egg, it creates a calcium pulse that goes through the cytoplasm, and that's what naturally triggers anaphase of a meiosis II spindle. And you can see here this movable loop. Here are some kinetochores. They're being pulled on, and finally they start moving. I'll let it loop one more time. So this is the meiotic spindle doing its job, and not inside a living egg now, but between a slide and a cover slip. And that was taken by a good friend, Paul Maddox, who was a real whiz with the spinning disk microscope. So the kinds of questions we've been asking, we and the other people studying the system, what signals trigger spindle assembly? I've been fascinated in how do microtubules behave in spindles. You could see from the introductory book, I'm very much a student of microtubule dynamics. And then how are the spindles spatially organized? And I'll rather briefly address those, give you some references if you want to get deeper into these. So first, what triggers spindle assembly? And this question of what regulates spindle assembly can be broken down into a time question, what triggers when the spindle occurs, and then a space question, where it occurs. And none of this is work from my lab. The time question, to trigger spindle assembly, the cytoplasm must enter a mitotic state, and this occurs by activation of CDK1 kinase, the CD2 cyclin B kinase. That's the master kinase that turns on all of mitosis. That's real textbook stuff, and it's by manipulating that system that we control whether the extract is in interphase or mitosis. The space question is really, why do you get a meiotic spindle here, right here in the egg, and not somewhere else? Why precisely in one place? And this is the exact north pole of the egg, that this spindle assembly. What's special about that place that makes the spindle form there and not anywhere else? And what's special about that place is that's where the DNA is, the maternal genome. That's where the condensed chromosomes, the maternal genome are. And what we now think is that the spindle forms around the DNA. And this was really the discovery and part of the life work of a good friend of mine, Eric Carcenti. Here's a couple of classic references. He used to, back actually when I was a graduate student, he was in Mark Kirshner's lab, and he was injecting DNA into eggs and watching what happened. And he noticed if you injected DNA, spindles would form around them ectopically. And he hypothesized all the way back in 1984 that DNA emitted some signal here, which he imagined would be a kind of gradient high at the DNA and declining elsewhere, that would trigger spindle assembly to happen right there at the top of the egg and not anywhere else. And for many years, the nature of that signal was completely mysterious, but now we know in molecular terms of at least 2 signals. And there was a beautiful experiment to prove that the DNA is the trigger was done by Rebecca Heald when she was a postdoc in Eric Carcenti's lab. She took a completely random DNA sequence from a bacteriophage, coupled it to plastic beads using magnetic beads that are convenient to manipulate, put those in the extract, and found that they triggered microtubule polymerization and spindle assembly. And there's one of their spindles again. So really just a classic proof of this Carcenti's concept that the DNA is the spatial trigger. I'll show you a movie of that taken here at Woods Hole by Aaron Groen and Tom Maresca in the egg extract system. This is the scale bars here, but I hope you can see here that this clump of circles are the DNA beads, and you can see the microtubules first form rather randomly around the beads and then gradually organize themselves, this is hours and seconds playing out here, into this characteristic bipolar structure. Now, this kind of spindle can't actually separate the beads. You know, unlike chromosomes, you can't split them in two and separate them, but we believe the actual assembly process, it uses all the same factors and is in many ways similar. And so our picture of self-organization of meiotic spindles is that you start with DNA assembled into chromosomes. We call that chromatin. That triggers microtubule nucleation near the DNA. The microtubules are then kind of sorted out and bundled, and motor proteins are important for that. I'll come to those in a little bit. But it's also very important, going back to the themes of my introduction, not to think of this as just a linear pathway, because once you have this spindle, assembly isn't finished, this structure is continuously rebuilding itself. So all the time, this same process is going on, so it's building itself continuously at steady state. Okay, so DNA is sending out some kind of signal. What is the nature of that signal? And I think Eric early on felt that it would likely be what we could call a reaction diffusion system. That's something with a local source stuck to DNA or stuck to chromatin that creates some unstable molecule that can diffuse away. That molecule is unstable because it's broken down here by a second enzyme. And if you have a reaction diffusion system like that, it'll give rise to a spatial gradient in concentration of the signal. And for those of you who are mathematically inclined, could figure out what the shape of this curve is, what mathematical function it would be for a point source. Here it suffices that the signal is high at the DNA and then decreases due to a combination of production, diffusion, and breakdown. And this concept is actually very similar to the concept of a morphogen in developmental biology, a molecule that diffuses to pattern an embryo. The difference here is instead of diffusing between cells, it's diffusing inside of a cell. And I think the spindle morphogens, if you want to use that term, diffusing away from DNA, is one of the best examples we have of a reaction diffusion system operating inside of a living cell. So I won't go into all the history of this. We actually now know of two signals emitted by DNA. One is a small GTPase Ran in the GTP form. Another is protein phosphorylation. The Ran signal is created by a Ran-GEF and then broken locally on the chromosome, and then a gap. This is a GTP exchange factor. This is a GTPase activating protein. The phosphorylation gradient is generated by Aurora B kinase and then opposed globally by phosphatases. There's a review for people who want to learn more about this. I'm only going to talk today about the Ran gradient, this one, but this phosphorylation gradient is also very important. I'm trying to keep it as simple as I can here. Here's a cartoon of the Ran system. The GTP exchange factor is localized on DNA. Ran.GTP, that's the inactive form of this small GTPase, then is switched catalytically. GTP is exchanged by RCC1. This is the active factor that triggers spindle assembly. Then the Ran.GTP is converted back to Ran.GTP by a GTPase activating protein that's global. And this was discovered by a bunch of people. I've put some names there. My lab was not one of them. I think this is absolutely fabulous biology. One other piece of information. This is the exact same system that powers nuclear import during interphase. So this is the system that powers NLS-dependent transport through nuclear pores, although I won't be talking about that today. And what Ran does, Ran.GTP, is there are important spindle assembly molecules. Here's one called TPEX2 that are sequestered. They're sequestered by binding to a molecule called importin, which is an alpha-beta heterodimer. So TPEX is inactive here because it's sequestered. Ran.GTP binds to importin beta and dissociates this complex into three parts. Now the TPEX is free to do its job, which is organizing spindles, and TPEX2 is actually one of the proteins Eric Carcenti worked on, my lab works on. We don't know precisely how it works. We do know it's a key spindle assembly factor. So this is the biochemistry. One question I think very naturally like to ask is, okay, I'm asserting there's a spatial gradient triggering that self-organization I showed you around the DNA beads, But can we actually see that spatial gradient? Can we visualize it? This is very difficult to do because, for example, if you fixed a cell for immunofluorescence, these are putatively soluble molecules. They probably all move around during your processing. And so this is a very difficult challenge. And this challenge was solved by Rebecca Heald and Carsten Weiss. Heald was a student of Carcenti's who did that DNA bead experiment. They built elegant FRET biosensors. This is fluorescence, resonance, transfer. And this is a kind of biosensor that people are using for a lot of applications in cell biology nowadays that allow us to see changes in the chemical state of the cytoplasm with a microscope. And the way this works is there's a Ran binding domain shown here in green. This is easier to see here. That's fused on its one N and C terminus. I'm not sure which is which to, although if I put the reference here, people are interested, to CFP and YFP. And when this molecule is on its own, CFP and YFP are close enough together to get efficient FRET, fluorescence resonance energy transfer, from the CFP to the YFP, so the FRET signal is high. That's if Ran is in the GTP form. If Ran is in the GTP form, it binds to the Ran binding domain, kind of separates the two halves of this molecule in space, and the FRET signal goes down. And this is one of two different probes that Heald and Weiss developed. And here is, I'll step back from this, here is their sensor doing its work. So this is a multicolor live imaging experiment. Here we're seeing the microtubules with fluorescent tubulin. Here we're seeing the YFP signal from the probe, here the CFP signal, and here the FRET signal. And this color here indicates this is a low value of FRET. If we go back, the low value, this is the high value, this is the low value, that's where the Ran.GTP is, where the FRET is turned off. And so you can see here this low value in the gradient indicates that there's a high concentration of Ran.GTP here. So here we're actually visualizing live in the extract this reaction diffusion gradient of Ran.GTP, which I just think is a really remarkable thing to see. And as I mentioned earlier, I think this is one of the best examples of a reaction diffusion gradient in cells. There are a lot of questions about this. One thing you'll notice is that the shape of the space in the extract, which has high Ran.GTP concentration, is not the same shape as the microtubules. So even although we believe this high Ran.GTP area here is triggering assembly of microtubules, we think it can't be dictating the exact shape of the microtubules or, for example, the exact length of the spindle. And how those are organized, I think, is going to come more out of the microtubules themselves and the motor proteins that I'll talk about in a bit. How do microtubules behave in the spindle? So it's the second topic I want to take up. So I talked in the introduction about how the spindle is continuously rebuilding itself, exchanging subunits with the cytoplasm. We've known that for a long time. But what is the nature of that continuous rebuilding? Just to give you a sense of the microtubules here, this is an electron micrograph of the spindle, and here's a kind of cartoon representation. One of the things I want to point out is that in these egg extract spindles, the vast majority of microtubules are of this kind here that don't attach to kinetochores, and only about 5% of the molecules actually attach to chromosomes. So when I'm talking about microtubule behavior using the kind of bulk measures I'm going to show, I'm talking about these so-called non-kinetochore microtubules. Those are the microtubules that give the spindle its size and shape. They're not the microtubules that actually segregate the chromosomes, but they're the most relevant microtubules, the bulk microtubules, for talking about this self-organization problem. So one thing we know is that microtubules turn over very rapidly, as I implied in the cartoon. A classic experiment that was done when I was still a PhD student by Ted Sam and Dick McIntosh and co-workers is a fluorescence photobreaching recovery experiment. This is a 1984, so this was done with a video camera, not modern digital camera, so it looks a little fuzzy. But the idea in this experiment is you have a spindle containing fluorescent tubulin. This is microinjected fluorescein-labeled tubulin. A laser was used to bleach one half of the spindle, and key to this experiment is you're not actually damaging the spindle, you're just turning off fluorescence. And then a few seconds later, you can see recovery here. And you can plot a curve of recovery, and in fact the half-time recovery is about 19 seconds. So Salmon McIntosh inferred that subunits are exchanging sufficiently rapidly that in 19 seconds, half the microtubules in the spindle have been turned over and refreshed. So even though the spindle can last hours, the subunits within it only persist for tens of seconds inside the spindle. And that really is a very characteristic of the spindle and important for those properties I talked about in the introductory material. I'll show you now, I've spent a long time and my co-workers studying microtubule behavior in spindles. I'll show you one recent experiment done by Dan Needleman and Aaron Groen, which is a single molecule imaging. So imaging has come a long way since that 1984 paper. Now here using a spinning disc confocal and a very sensitive camera, we can see these dots here actually, these turn out to be single tubulin molecules. And what we've done here is we've spiked a very low concentration of fluorescent tubulin into the spindle, such that only 1 in every 200,000 molecules approximately is fluorescent. That means a single microtubule on average is only going to have 1 fluorescent spot in it. And if I step back and play the movie, I hope you can see here these dots. Usually our eyes are most attracted to the fact that these spots are moving. They're all sliding. If you look carefully, the ones at the in the southern half are sliding south, the ones in the northern half are sliding north. But if you look carefully at each dot and try to follow it, you'll notice it will disappear or a new one will come up. And our interpretation of that movie is shown here. So when a tubulin molecule is just diffusing around in solution, in cells approximately half the tubulin typically is in a somatic cell is soluble, and about half is in polymer. Here in the extract, the vast majority is soluble. A soluble tubulin molecule is diffusing around very quickly. It takes about 500 milliseconds to acquire an image on the camera. In that time, the soluble molecule will diffuse around and just blur out. If that molecule is kind of captured by a growing microtubule, it now moves much more slowly and we can image it. And we can image it for as long as it's in the microtubule. When the microtubule depolymerizes again, it goes back to being soluble. So we can, I've just made that point here, but we can actually measure the lifetime in polymer of a single tubulin molecule by seeing how long we can track it for. And then if you watch the spot moving, I've shown here moving towards me, we can also track the whole microtubule sliding. It's a kind of fiducial mark, if you will, for the microtubule sliding. It's easier to see the turnover, the single spots coming and going if you block the sliding. And in this experiment, the sliding was blocked with a drug that freezes kinesin 5, a motor in the spindle I'll talk about. Now the spots aren't sliding, you can see them almost twinkling. What that twinkling is each spot comes, persists for a few seconds, and then disappears again. And this experiment actually also shows that you can uncouple sliding from polymerization dynamics. Those spots are sufficiently bright and well separated that they can be tracked by automatic software. This is Dan Needleman's work using software taken from physics labs who study particles moving. You can see here trajectories, these vertical lines. This is a control spindle. This is one of these drug-frozen spindles, so everything's frozen there. And using that kind of software, you can quantify both the sliding and also how long each spot persists, which is the lifetime of that spot in polymer. And here's a lifetime plot. So here's how long the spot can be imaged for versus the fraction of spots. And there's about 16,000 individual spots went into this graph here. And the blue spots are data here. The red line here actually is a fit to a theoretical curve, t to the minus 3 halves, e to the minus t over tau. There's one adjustable parameter, this a tau parameter. And then the average lifetime here is 16 seconds, so the midpoint of this distribution. Couple of points here. This 16 seconds is, I hope you recognize, remarkably similar to that classic Salmon and McIntosh number in sea urchins. So this rate of turnover with this more sophisticated modern method really confirms roughly the old value. This equation here, I have to say this is the most complicated equation I've ever run into in my own work, and it took me a long time to understand this. What this equation actually is, if I go back to this slide here, this is the first passage time for a one-dimensional random walk. It's the expected time if the tip of the microtubule is doing a random walk in position. First passage time is coming from the red spot back to the red spot. So this is exactly the curve here that you would expect if the plus ends of the microtubules are doing a random walk. And this dynamic instability process, these large length fluctuations, we believe is a form of random walk. So this is a satisfying match to theory for what to me is a fairly complicated equation. I would say it's not quite that simple, and if people are more interested in the quantitative, we had a lot of arguments with the reviewers of the paper about how to precisely interpret that, and I would refer people to Dan Needleman's paper in NBC in 2010 if they're interested in that. So summarizing a lot of work of that kind, the picture of the spindle that emerges is here, where the yellow bars are some kind of nucleating structure, the green is the microtubule, and the white arrows are the sliding movement. So microtubules are nucleated throughout the spindle. Their plus ends grow and shrink by dynamic instability, which has a random walk-like character. On average, they live 16 seconds and grow to about 5 microns in length. And then they slide polewards mostly at variable rates. So there's this sliding phenomenon. So the microtubules are quite a bit shorter on average than the spindle itself, and they're all sliding. So this, I think, matches now this picture I gave in the introduction of the spindle continuously renewing itself and shows us how the microtubules are behaving. It leads, of course, to a lot of other questions. One question we've been very interested in, how are these short microtubules organized? Why are they all sliding? And I'll give you some answer to that. And then how are they nucleated? And what are these yellow boxes here that are initiating the microtubules? That's an unsolved problem that we and others are working on at the moment. For the organization and sliding, we know there's a very important role for motor proteins, ATPase motor proteins, which I'll introduce a bit more in a second. We know that mainly from inhibition or depletion experiments. Here's a series of inhibition experiments that were done here at Woods Hole in 2000. Here's some control spindles by polarization microscopy. Here we've inhibited the minus end-directed motor cytoplasmic dynein, and hope you can see that the poles, instead of being focused as in the controls, are all kind of splayed out. It's very clear, I hope, in that one. Here we've inhibited a plus end-directed kinesin I'll talk about in a second called EG5 or kinesin 5. Now the poles have collapsed together. This shape here is a radial array of microtubules or aster. Fascinatingly, and we don't actually fully understand this at all, if you inhibit both motors, the structure sort of fixes itself. It looks a bit like control, or these are much more fragile. They fall apart more easily, especially if you poke them. And that kind of experiment gave us a picture of the spindle like this, where the minus end-directed motors help cluster the poles together. So if you get rid of them, the pole slays out. And the plus end-directed motors are helping keep the spindle bipolar. We spend a lot of time on the plus end-directed motor in this problem, kinesin 5. It's a plus end-directed tetrameric kinesin, and homologs of this motor are required for spindle assembly in most eukaryotes, going down all the way to yeast and up to plants. I'm cartooning it like this. These are the 4 motor domains, and there's 2 coils that interact with each other in an anti-parallel fashion. Here's a cartoon of it. Here's a movie of kinesin walking along microtubules from my colleague Ron Vale and his lab, based on crystalline EM structures. And so this is drawn for conventional kinesin, but if you imagine this being one end of EG5, and then the other end being up here. And the 2 ends are the same, so of course it's appealing to hypothesize that they interact with 2 different microtubules and cross-bridge, and that idea was first proposed by John Scoley quite a while ago, a colleague in the field. And one reason we've been able to work on kinesin 5 in our spindle system fairly easily is we have some nice small molecule inhibitors, drugs. And these drugs bind. This is the ATP binding pocket in the head of kinesin 5. This is a nice drug binding pocket. The first drug that we actually found that fits into that pocket is a drug called monastrol. which we found by chemical screening back in 1999. Since then, much more potent and specific inhibitors have been developed by industry groups in the hope of their being useful for treatment of cancer patients. And that actually is a whole story. Our kinesin 5 inhibitors have not been successful yet in treating cancer, and why is that, I think, is an interesting story that I'm not going to talk about today. These class of drugs trap the motor in a state with weak affinity for the microtubule, so it's like removing the motor. And we can do the kind of experiment shown here. Here's a control spindle where this kind of speckles here, you can see the sliding of the microtubule. Here's the same kind of spindle treated with a kinesin 5 inhibitor, and if I step back, I hope you can see the microtubules are sliding apart continuously in this one, whereas here they're static. And from that kind of movie, the image we get is this one, very much similar to John Scully's original hypothesis. The kinesin 5 is bridging two anti-parallel microtubules here. This end is walking towards the plus ends, that end's walking towards the plus ends. That's creating a force on the microtubules, shown in green, that slides them apart and kind of pushes the two poles of the spindle apart. And that's a current view of how kinesin 5 works in the field. And so, here's this kind of motor-driven sliding field, and I want to relate that to the image I showed in the introduction, where I talked about the poles being pushed and pulled, and now bring in specific molecules. So the dynein is kind of focusing the poles together and pulling them inwards. The kinesin 5 is pushing them out. And those motors are coming and going from the spindles. They just like tubulin, they turn over very quickly. And so the spindle is continuously repositioning the motors and rebalancing the forces. And again, this is part of this kind of dynamical steady state that gives the spindle its lifelike properties. So a lot of details. I hope that was interesting. Let me quickly summarize them. Egg meiotic spindles self-organize in the vicinity of DNA triggered by a reaction diffusion of Ran.GTP and also a kinase gradient. They're built of relatively short microtubules whose plus ends undergo continuous dynamic instability. They're organized by these motor proteins, plus and minus directed motors that promote sliding and clustering. And what I would argue, although we don't fully understand it, is together these reactions generate this very dynamic steady state in size and shape that's continuously rebuilding itself, continuously adjusting the forces so they balance, and that's what gives these very lifelike forces. So if we go back to this movie I showed in the introduction of two spindles fusing, which to me, more than anything, sort of shows the strange properties of this structure compared, for example, to the bacteriophage, which certainly wouldn't do this. I hope that kind of more molecular picture can help you begin to frame how this works, although I do not want to pretend that this is a solved problem by any means. There are many future questions, particularly how microtubules are nucleated. The size and shape of the spindle are very much governed by where microtubules are nucleated, and that's an unsolved problem biochemically. We know it's downstream of that Ran gradient, but what it is in molecular terms, we don't know. We would like, and we're going to certainly need some help from physicists and mathematicians here, to develop a quantitative understanding so we can actually explain size and shape, and also time scales in quantitative terms. And I'll leave you with one last point here, is interphase microtubules. So if you change the cell cycle, everything changes. And for many years we just had only focused on mitosis. Now we've gotten very interested in interphase, and that's a whole seminar, but let me just give you one snippet here. This is the work of Anna Ewan and Christine Field. And this is similar kind of microscopy, lower magnification. This is very low mag. This whole field is 600 by 800 microns, so almost a millimeter. You can see in blue here are sperm nuclei, in green are microtubules. And what we're doing here is we're mimicking fertilization. We're putting in sperm and calcium at the same time to make the extract go into interphase. So the sperm are going to build this big aster that they use for organizing the egg that's similar to that zebrafish aster that I showed you very early on in the introduction. And what you'll see when this movie runs is these asters can move and they can solve a kind of geometric problem of spacing themselves out. So I'll stand back a little. So you can see the microtubules growing out, interacting here, plus end to plus end, and I hope you can see that they're actually solving a fascinating geometric problem of spacing themselves out evenly in the field. And how that works and the role in the biology of fertilization and early development is a whole other topic. And if I want to leave you something there, it's like there's really still many cool self-organization problems left to study. So let me come back to a slide that I used in my introduction here, where this complex and beautiful bacteriophage that has an invariant size and shape that's kind of directly encoded by genes. The meiotic spindle I've spent a lot of time talking about now, where a self-organization pathway leads to a much more dynamical assembly that's at steady state, continuously rebuilding itself, continuously rebalancing forces, and that gives it the ability to adapt in size, to repair damage, and these other kind of lifelike properties. Both beautiful, but somewhat different in principle. And to me, this one is really alive. I hope I can use that word. And let me finally end by thanking the many wonderful students and postdocs I've worked with. I didn't make a list. I hope I mentioned many of them by name in the talks, particularly my good friend Ted Salmon, who I've worked with at MBL for many years as the MBL Cell Division Group, showed a number of movies taken here at the MBL. Also inspiring colleagues, particularly Mark Kirshner, my PhD advisor, Eric Carcenti, who discovered the idea of DNA organizers, and Shinya Inoue, who really one of the early and most thoughtful students of self-organization. NIHGM, who funded most of this work. They are the main supporters of basic biological research in the US. GoGM, they turned 50, I think, last year. And finally, of course, the frogs who made all this possible. Thank you.